his PhD students who settled in abroad and as well as other universities in Karnataka and India. And you can see 3 of his students have been working as professors at the Department of Biology University, which is a rare feat. And all of them, they have published wonderful research articles. It's because of the able guidance of Professor K. Kemparaju. Without wasting much time, I request Professor K. Kemparaju to take on the dais and deliver his talk saying the neutrophils, the sentinels of innate immunity. Thank you all.
Thank you, Kumar, for the nice introduction. In fact, it is too much for me. He said too much. Anyway, I take this opportunity to thank Professor Kumar for this invitation. I also thank all the faculty members, Mrs. Kumar, Santosh, and everyone present here. My group at the Department of Biochemistry, University of Mysuru is working on various aspects of snake venom pharmacodynamics. These are the different areas we work on. But in this class, I'm not going to present on any of these concepts, mechanism of hyperglycemia, sustained instruction, mechanism of specific hypoxia, and there are so many aspects to work on. But in this class, I'm not going to present any of these aspects. Instead, what I do is, I would like to randomly using some of the data that we have generated from our laboratory in order to justify this title. This title is Neutrophils, the sentinels of innate immunity. The sentinels means a kind of caretaker, a kind of guardian. So with this, I can replace the title as neutrophils as the guardians of innate immunity. The neutrophils as the caretakers of innate immunity. That's the meaning of that. Am I audible? Am I audible? So let me start my talk with a wonderful, thought-provoking statement from the Bible. That is, the life of the flesh is in the blood. Indeed, it's a true, very true scientific statement. Because if you analyze, understand, integrate biochemistry and physiology holistically, whatever research we do in animal system or cell culture system, they are fall in the framework of this statement. The life of the flesh is in the blood. Meaning, no blood means no life for flesh. It's as simple as that. So, in majority of the major accidents, people die. It's not because of the organ failure, because of lack of some organ. It is mainly because of the loss of blood. So now you can imagine the significance of the blood in making the rest of the tissues alive. So if you sacrifice an organism, removing its blood, then that process is what we call exsanguination. So blood being the liquid connective tissue, it performs such vital functions as maintaining hemostasis, transportation of various molecules, and play a major role in defense. Hemostasis means regulating the concentrations of various molecules, various ions in the body, and also maintaining the body temperature. Transportation is, so in this case, blood is the one that transports oxygen and nutrients to various tissues and organs, and at the same time bring back the toxic metabolic waste and carbon dioxide from sites of their origin to the sites of their disposal. And most importantly, blood play an important role in the defense, the resistance against the dangerous foreign invaders. So if you look at the defense, we have various systems, very complex and complicated systems are working in the blood. Let's say, for example, clotting system. Clotting system. So we know, like we have in clotting, we have various pathways operating: extrinsic pathway, intrinsic pathway, and we have common pathways, and all of them end up in converting prothrombin to thrombin. Thrombin is the one that converts fibrinogen into fibrin. So in this case, the striking thing is that each factor is activating the other factor through limited proteolysis. Is it not? So the most important striking event that you can notice in this case is that fibrinogen, although it's a large molecular weight structure, once it is subjected to limited proteolysis, you can see a drastic change in its property, where the fibrinogen is soluble. Once it is converted into fibrin through limited proteolysis by removing alpha peptide and A peptide and B peptide, it becomes insoluble. So we have a fibrin clot formation. So in this case, what happens is, whenever there is a damage to the tissue or the blood vessel, immediately platelets get activated and they adhere to the damaged site. Right on top, there will be formation of clot. So it seals of the damaged site. It's something like platelets act like bricks over which we have the clot, we have cement, a temporary wall is created. So that's how the blood loss is prevented. So clotting mechanism is most important defense mechanism. And followed by we have complement system. Complement system represents a system of heat and serum proteins. So in this case also we have various pathways are operating. We have classical pathway, alternate pathway, lectin pathway, we have thrombin pathway, helictin pathway, and so many other pathways are working. So in this case also, whichever the pathway is activated, the end result is the same. So the end result is the formation of membrane attack complex at the end. Is it not? Membrane attack complex is the one that gives a lethal hit to the target tissue. So thus the clotting system and complement system play an important role in innate immunity. And in addition, we have various cells participating in innate immunity. So if you take into account the cells, we have, some of the cells participate exclusively in innate immunity, some of the cells participate exclusively in acquired immunity, some of the cells participate both in innate immunity and acquired immunity, and some cells bridge both innate immunity and acquired immunity. So among these cells, neutrophils play an important role. They participate both in innate immunity and acquired immunity. So this is the cartoon showing the different stages of development of neutrophils and stage-specific formation of granules called neutrophil granules. So you can see over here, these are the different stages of neutrophil development. Start from promyelocyte, and finally we have the matured terminally divided cells, the neutrophils. So in this case, during the development, you can see the formation of various granules. So the first granule that are being made during the development is the eosinophilic granule, what we call primary granule. And after that, it gets into the next stage. Next step, myelocyte. So when it gets to the next stage, the formation of primary granules or different granules ceases. Then keep it as keep it as So we have the other granule formation take place, or we have secondary granules, otherwise called specific granules. And then in the next step, we have the the tertiary granules, what we call the gelatinous granules, and finally we have the secretory vesicles are formed. That means if you take the neutrophils, neutrophils are loaded with 4 types of different granules. There are the primary granules, secondary granules, tertiary granules, and secretory vesicles. All these granules are loaded with lots and lots of antimicrobial, antibacterial, cytotoxic agents. So in primary granules, these are the various antibacterial or antimicrobial or cytotoxic agents are present. Neutrophil elastase, myeloperoxidase, ketapsin G, proteinase 3, lysozyme, beta glucuronidase, alpha deficiency. There are so many. And secondary granules, keep it around this later. So, secondary granules are loaded with these antimicrobial and cytotoxic agents. Tertiary granules are loaded with these, and secondary vesicles are loaded with. So finally, if you take the neutrophils, the mature neutrophils, otherwise called granular cells, they are loaded with lots and lots of the granules, which contains lots and lots of antimicrobial, antibacterial, cytotoxic agents. So different groups have discovered variety of antibacterial cytotoxic agents. There are 40 different antimicrobial or antibacterial agents have been discovered, known to be present in these granules. So as the neutrophils develop, the granular size decreases while the degranulation propensity increases. So in response to stimulation, these neutrophils undergo degranulation. They release their granular content. So all these granular contents are released. So why today I'm discussing this is because the neutrophils perform a different function. So let's say if you take neutrophils are known to mount 3 levels of defense. They are the phagocytosis, that is engulfing and digesting the particulate material. If the neutrophils are unable to carry out phagocytosis, then they move on to the next level of defense. They right on top of the foreign invader, they carry out, they undergo degranulation. That means they release their granular content. So as I said, neutrophils are loaded with lots of granules and they contain cytotoxic or antimicrobial agents. So if they cannot phagocytose foreign structure, if the foreign structure is larger enough, in that case, what happens is they just congregate on surface of the foreign structure and they undergo degranulation, release lot of cytotoxic agent and tries to kill the foreign invader. When the neutrophils got to know that phagocytosis and degranulation are not going to work, then they mount a different level of defense, an ultimate level of defense, where finally they work like soldiers, they are ready to die for the defense cause by injecting their nuclear content through a process what we call mitosis. So just I wanted to highlight in this talk the other defense process performed by the neutrophils. So phagocytosis is very familiar, everybody aware of phagocytosis, and also degranulation. But mitosis is not featured in the textbook. That's the reason why I am just highlighting the importance of neutrophils in net formation. Mitosis is an ancient bioweapon in innate immunity where neutrophils ejected decondensed DNA, decondensed chromatin, put the nets means neutrophil extracellular trap, coated with lots and lots of antimicrobial, antibacterial or cytotoxic agents. This is the cartoon showing one neutrophil is ejecting the decondensed DNA. So you can see over here, the decondensed chromatin, decondensed DNA is ejected like that, and it is coated with lots and lots of antimicrobial and antibacterial and cytotoxic agents like this. See, you can see only one neutrophil is ejecting the decondensed DNA. Now you just imagine 10 neutrophils, 100 neutrophils, 1000 neutrophils, lakhs of neutrophils are simultaneously ejecting the decondensed DNA. Now you can imagine the impact of the nets decondensed DNA, the chromatin that is being released. So if you imagine that the final nets, it is something like the nets formed by so many number of neutrophils. It's going to be like a thick spider web or a kind of miniature pattern that can like any blood vessel and prevent the blood flow. See, it's something like this. See, if you take the human genome is actually made up of about 3 billion base pairs. So 3 billion base pairs, 1 billion means 100 crores. So 3 billion base pairs are involved in making 23 chromosomes. So whenever we talk about the genome size, in case of human genome, we take the haploid genome size. So haploid genome, we have 23 chromosomes. So 23 chromosomes are made out of 3 billion base pairs, is it not? So if you take the diploid cell, the neutrophils being diploid, we have 46 chromosomes. That means the size is doubled, is it not? It's made up of, I think, over 6 billion base pairs. 6 billion base pairs of nucleotides are involved in making about 46 pieces of chromosomes, is it not? If you take all 46 pieces of chromosomes, so unwind the DNA and place one after the other, and if you measure the distance or the length, it comes to around 2 meter long. So that means each neutrophil is able to eject about 2 meter long DNA. So now just imagine one neutrophil is ejecting 2 meter long DNA. Let's say 10 neutrophils, 100 neutrophils, 1000 neutrophils, 10,000, 1 lakh neutrophils are simultaneously ejecting about 2 meter long DNA. Just imagine the impact of the net. So that's what the net is. That is, it can form by web-like structure or a thick, cotton-like structure. You can see over here. So this novel mechanism of netosis was discovered by Walter Brinkmann and Arthur Zisinski from Max Planck Institute, Germany. And they published their novel discovery in a top-notch journal, Science, in 2004. See, the whole story of netosis was started with these findings. So, way back in 1980, this was the first report reported by M.S. Fan from John Hopkins Institute, Baltimore, Maryland, USA. So, 4-bar myristate acetate induced neutrophil autophagy. This was the first report where he reported the 4-bar myristate acetate induced neutrophil autotoxicity. PMA, 4-bar myristate acetate means it is PMA. Is it not? You are aware of the PMA, is it not? PMA. Are you aware of PMA? 4-bar myristate acetate. So this is actually an activator protein kinase C. So it's a non-hydrolyzable activator protein kinase C. So protein kinase C activates neutrophils and cause the death of neutrophils. That was the first observation way back in 1980. And then one more study came from a group from Portugal, that is activation of human neutrophils by 4-bar esters decreases cytoplasm compactness and the lactopherrin content of granulocyte. See, this another study came in, and followed by one more study, rapid killing of human neutrophils by the potent activator 4-bar myristate acetate, that's PMA, accompanied by changes different from typical apoptosis or necrosis from a Japan group. See, you can see over here. See here, the PMA induced death of neutrophils was different from apoptosis or necrosis. Apoptosis. So, way back in 1996, before 1996, apoptosis or necrosis mechanisms were understood. And here, they are observing the PMA-induced death of neutrophils was different from apoptosis and necrosis. Apoptosis and necrosis. So, and when people wanted to understand what could be the mechanism of PME-induced death of neutrophils. So in 2004, it is these people demonstrated that PME induced the death of neutrophils through a mechanism what we call netosis. So this is what netosis means. So various pathogens would induce net formation. These are the various pathogens studied for net induced, like these are all particulate structures. Not only particulate structures, even the so this is how this is the SEM of scanning electron micrograph of salmonella typhi caught in the net. So these are the salmonella typhi caught in the nets. These are the nets. This is how it was trapped. This is SEM of Scissor flex negative. This is another microbe. This is caught in the net. So this microbe caught in the net. This is the picture from publication from Benjamin only. So not necessarily the particulate structures, even the soluble structures can induce net formation. Various soluble structures, for example, PMA, 4-barbitol acetate, interleukin A, tumor necrosis factor alpha, GM-CSF, that is granulocyte monocyte colony stimulating factor, along with C5A. C5A, it's a component formed for complement activation. GM-CSF plus LPS, like polysaccharide. So LPS alone, platelet activating factor, hydrogen peroxide, nitric oxide, T-selectin, statins, and various venom toxins can induce net formation. Soluble factors can also induce net formation. That's what the bottom line is. So, as I said, my group is working extensively to understand the molecular mechanisms involved in sustained tissue destruction caused by viper bites. So this is how the local toxicity is caused. So African Echis ocellatus venom caused like this, and Vitis erythemas eriadans also cause local toxicity like this. See, in the Indian subcontinent, we have the big four snakes. We have cobra, crane, Brussels viper, and Sasquatch viper. Of the four, these two, Brussels viper and Sasquatch viper, are known to inflict this kind of local toxicity. They continue tissue destruction like this. However, the molecular mechanism of how the local toxicity is caused was not clearly understood. However, the only clue was the accumulation of lots and lots of neutrophils at the venom injected site. So we wanted to repeat that experiment in our laboratory. So we did that experiment in our laboratory using mouse tail model. This is the mouse tail tissue section. You can see clearly over here the muscular striations characteristic of skeletal muscle over here. This is the control tissue, and this is the venom treated tissue section. Here there are no clues for presence of muscular striations. The entire tissue looks like a mixed tissue. But if you carefully observe the tissue, you can see over here the entire tissue is studded with or loaded with black or purple particles. And if you take this zone and blow it up, it looks like this. So now here the black or purple particles are more conspicuous, more visible. So these are actually nothing but the polymorphonuclear leukocytes or the neutrophils. You can see here the Echis venom in the Indian subcontinent induce local toxicity. In that event, it will recruit lot of neutrophils at the site of venom injection. So this gave a lead for us to speculate whether neutrophils play a role in sustained tissue destruction or not. So in order to understand that, what we did was we we isolated neutrophils, and these are the control neutrophils. These are the control neutrophils. You can see over here. It is stained with blue stain. See, neutrophils are very characteristic in a way that they possess multi-lobe nucleus. Otherwise, rest of the cells possess actually circular or round nucleus. Neutrophils possess the multi-lobe nucleus. See, especially this stained part is just nucleus is stained here. It is stained using blue stain. So this nuclear part is stained. So nucleus is actually multi-lobe here. So that's the reason why the neutrophils are also called polymorphonuclear leukocytes because it contains multi-lobe nucleus. And this was found by the discoverer of the phagocytosis, L.A. And neutrophils, the name is given by polarly based on the staining. So based on the staining, they are called neutrophils called by polarly based on the nucleus shape. Elimination combined cells as polymorphonuclear leukocytes. So these are the control neutrophils. You can see clearly the neutrophils over here with multiple nucleus, and we treated neutrophils. This is the that's black and white. This is black and white. This is the same one. This is colored one. So this is after venom treatment. This is you can see over here with 5 microgram HSCan and a venom. So neutrophils are now programmed for actually netosis. They are ready to eject their DNA. See, the multi-lobe characteristic nucleus is lost, and the nucleus has taken up the round shape, and some of the cells have already ejected their DNA. Color. This is with the next concentration, about 10 microgram concentration. Color. See, 25 microgram concentration. So many of the cells have already ejected their DNA, decondensed DNA. This is the colored one. This is a 50 microgram per ml concentration. You can see almost all of the neutrophils have ejected the DNA. Now, see, look at this. It looks like a thick spider web are put together. It's like a miniature cotton flag. You can plug any blood vessel. That is the impact of the net formation or netosis. Colored one. See, this is the positive control. For any experiment, you have to keep a positive control. So in this case, we kept PMA, 4-ball crystal acetate, as positive control. So this PMA can induce netosis at nanomolar concentration over here. So you can see over here, so at 50 nanomolar concentration, it can induce netosis massively. So PMA can readily induce net formation, stimulate neutrophils for netosis. So if you want to assay neutrophils, so netosis, what are the markers you can think of? So you can assay decondensed DNA and chromatin fibers, and you can assay, most importantly, citrullinated histone 3 or X3CAT, or neutrophil elastase, myeloperoxidase, PAD4 enzyme. PAD4 means peptidine arginine deaminase 4 isoform, not aminase. It is deaminase 4 isoform. You can also say actin dynamis. You can also say cathepsin G. You can also say lactoferrin. See, remember while explaining the net formation, neutrophils are loaded with granules, 4 types of granules. Those granules contain lot of antimicrobial or cytotoxic agents. So during netosis, there is degranulation take place. From the granules, the antimicrobial agents comes out, and they put the DNA. Isn't it? They put the DNA. So thus, so you can see over here, all these cytotoxic agents and antimicrobial agents have coated the DNA, ejected DNA fibers. So you can use them as markers to assay neutrophils here. So dependency DNA can be assayed directly. Acetylated histone 3 significance I tell you. Neutrophil elastase from the eosinophil granules and myeloperoxidase from eosinophil granules, that's also released. And PAD4 enzyme, peptide arginine D minus 4 enzyme, you can assay actin dynamics and so on and so forth. See here, this is how we assayed the necrosis. We took neutrophils, treated with venom or PMA, incubated for 180 minutes, centrifuged, took the supernatant. Supernatant contains neutrophil elastase, myeloperoxidase, coated DNA complex. This is the DNA complex, the supernatant. So we have this is the dose dependent effect, this is the time dependent effect, this is the principal endpoint here. So we have anti-MPO monoclonal antibody was coated to plates here, because we have MPO coated here. MPO coated. So neutrophil elastase and myeloperoxidase, they always work together. They coat together to the DNA here, release DNA. So myeloperoxidase is there here. So that's why we use anti-myeloperoxidase monoclonal antibody here coated. We have here the supernatant contain myeloperoxidase, all those things here, and we have used anti-DNA monoclonal antibody coupled to HR. This is how we measure or quantitated the next release. See, I said neutrophil elastase and PAD4 enzyme. These are important for net formation. See, what happens is neutrophil elastase is actually always associated with myeloperoxidase, and this neutrophil elastase is actually residing in the eosinophilic granules, that is primary granules. When the neutrophils, undergo degranulation, neutrophil elastase comes out. Okay, I'll explain here. See, to explain that, let me take the molecular mechanism here. See, the necrosis started with the PMA induced death of neutrophils. Remember, in the beginning, Emerson showed neutrophil autotoxicity, which was induced by PMA, and later it was discovered that PMA induced necrosis by Walker Brinkman and other physicists. How PMA induced necrosis is that the PMA is an activator of PKC. PKC, protein kinase, which is a serine and threonine kinase, it's a cytosolic enzyme, and it is activated by normally diacylglycerol and calcium after PI turnover, is it not? So PMA can directly activate PKC. So, but the natural activators are diacylglycerol and calcium. But without the requirement of diacylglycerol or calcium, PMA can directly activate protein kinase enzyme. So PKC, which is then activate a NOx enzyme. NOx means NADPH oxidase or carbohydrate oxidase. This enzyme is responsible for making ROS. What NOx does is it partially reduces the oxygen and convert oxygen into superoxide radical. So if you take oxygen, so if it is reduced by 1 electron, you get superoxide radical. If it is reduced by 2 electrons, you get hydrogen peroxide. If it is reduced by 4 electrons, you get what do you get? You get water, is it not? So in this case, NOx enzyme convert oxygen into superoxide radical, which is ROS, which is immediately converted into H2O2, hydrogen peroxide. So in an ROS dependent manner, neutrophil elastase and myeloperoxidase translocate out of the eosinophilic granules into the cytosol, and then from the cytosol it gets into the nucleus, it translocates into the nucleus. In the nucleus, neutrophil elastase, which is a serine protease, so what happens is this neutrophil elastase specifically degrades H4, histone 4. In the nucleus is loaded with a compactly packed with DNA and histone, is it not? So we have nucleosome here. nucleosome is formed with the help of histones and the DNA, is it not? So DNA is compactly held by histones. We have various histones, H2A, H2B, H4 and H3. Here we have nucleosome. So what neutrophil elastase does is it degrades H4 preferentially. After degrading H4, it would degrade other histones also. As a result, what happens is, see, when histones are gone, now what happens is, because DNA and histone are held by non-polar interaction through electrostatic interaction. Here, the protein is degraded. As a result, what happens is DNA unwinding takes place. Because if the protein is lost, we have negative charge on DNA increases, negative charge, refilling takes place, unwinding takes place. As a result, see, decondensation of DNA takes place. This is how neutrophil elastase cause DNA decondensation. As a result, it triggers the expulsion of nets. This is one mechanism where neutrophil elastase play an important role. So, along with myeloperoxidase, it does this job. So, we don't know why. If you knock off neutrophil elastase, myeloperoxidase activity is lost. If you knock off myeloperoxidase, neutrophil elastase activity is lost. Together they work here. Okay? So neutrophil elastase degrade the histone, as a result promote DNA unwinding, so leading to expulsion of nets. See, here what happens is, when the neutrophil elastase comes out of the eosinophilic granules, when it gets into the cytosol, see what happens is, earlier studies they have demonstrated that neutrophil elastase, before entering into, let's say, nucleus, it degrades the F active. So F active dynamics is altered in this case. F active dynamics is very critical for the stretching of pseudopodia or the mobility of neutrophils from one place to a different place. So neutrophils can easily move to a place where its work is required, role is required. So through chemotactic attraction. So for mobility, F-actin dynamics is very essential. So F-actin and G-actin will polymerize into F-actin, is it not? So through that, F-actin, G-actin polymerization, depolymerization, pseudopodia stretch like this, is it not? So here, what happens is, in order to keep the neutrophils at place where neutrophils is required, the F-actin dynamics is altered, and that is dynamics is inhibited, or neutrophil elastase is found to degrade F-actin. As a result, F-actin dynamics is altered. As a result, what happens is neutrophils remain arrested at the place where mitosis is required. That is a critical one. See, this is how neutrophil elastase play a role in mitosis. This is one. That's why I said neutrophil elastase is an important marker for net formation. Neutrophil elastase play a role in decondensing decondensation of DNA or net formation. See, in addition, PMA can also induce endoplasmic reticulum stress. As a result of endoplasmic reticulum stress, we have calcium ions are ejected out. So intracellular calcium generally stored in the endoplasmic reticulum, smooth endoplasmic reticulum. However, mitochondria also stores a lot of calcium, but the mobilization of calcium from the endoplasmic reticulum is very well understood. So, PMA can induce stress in endoplasmic reticulum. As a result, you can see an increase in intracellular calcium ion concentration. As a calcium ion dependent manner, this PAD4 enzyme, peptidyl arginine deaminase 4, isoform 4, is actually getting activated. So this PAD4 enzyme gets into the translocated into the nucleus, and its main target is H3, histone 3. So it catalyzes this reaction. What it does is it catalyzes the hydrolytic deamination of peptidyl arginine. and it converts peptidyl arginine into peptide citrulline. See, hydrolytic deamination means, see, look at this. It's this is let's say histone 3. We have arginine. Arginine has the side chain guanidinium group here. So the arginine is actually basic amino acid. It's an important basic amino acid. And why it is basic is because it contains a side chain guanidinium group. And this guanidinium group carries positive charge at neutral pH. And at neutral pH, you know, it carries positive charge and it holds the negative charge of DNA. So that's how DNA and histone, they make electrostatic interaction and they make a complex here. So what this enzyme, peptidyl arginine deaminase, so does is it converts the arginine into citrulline here. As a result, what happens is, you can see over here, the positive charge here, there is no positive charge. So loss of positive charge takes place. As a result of loss of positive charge, you know, there is no positive charge to hold the DNA. As a result, negative charge on DNA increases. So negative charge in a rippling takes place. That would also cause, you know, decondensation of DNA. This is all. So PMA will cause endoplasmic reticulum stress, calcium rise, in a calcium ion dependent manner, PAD4 gets into the nucleus and act on its preferential H3, and later other histones cause decondensation resulting in net formation. Netosis. This is how the mechanism of netosis works. See, this modification in other proteins is responsible for epigenetic modification. This is also an example for epigenetic modification. So this epigenetic modification, you know, it would eventually lead to autoimmune disorder as well. Because this is the normal component of the protein, all proteins, arginine. So look at this here, citrulline is not actually coated by any codon, is it not? Now it is present in protein. It may serve as an antigen. So against which, you know, antibodies are made, maybe leading to autoimmune disorder. Thus, H3, histone 3 is converted into citrullinated histone 3. This is how citrullinated histone 3 can also be a marker for assaying netosis. We can assay the formation of H3CAT as an indication of net formation. So I also said here, so neutrophil elastase, before it entering into nucleus, you know, it affects the effective dynamics. But how exactly effective dynamics is altered, we have not understood the molecular mechanism. But see, you can see over here, just the experiment. See, here we try to demonstrate the effect, the altering of effective dynamics in this picture. See, these are the normal neutrophils, confocal images. You can see clearly the nucleus stained, multilobe nucleus, so who stained? And this is, the red one is actually defective, polymerized the effective red one. And these are control cells. You can see a lot of signal for the red signal indicating the effective, and this is the principal employed. And here we used a toxin isolated from cobra venom, what we call cardiotoxin, for this purpose. So you can see over here, cardiotoxin induced massive net formation. Here you can see decondensation takes place here, and the disappearance of red color here. The red color disappearance means it's affecting active dynamics. So you can see with this is with 10 minutes incubation, and this is control cells, 10-minute incubation, and this is a 30-minute incubation. You can see disappearance of red color and ejection of nets. And with 60 minutes incubation, you can see almost red color has been disappeared and the cells have ejected the DNA. So in this case, you can see over here, cardiotoxin, it altered the active dynamics, and in presence of protease inhibitors like neutrophil elastase inhibitors, serine protease inhibitors, metalloprotease inhibitors. So in presence of inhibitors, there is disappearance of the effective. That means it indicates that effective dynamics is getting altered. This alteration is not because of proteolysis, it is because of disassembly. Okay? So these are the control cells. So if you use the active polymerizing and depolymerizing inhibitors, you don't see any, netosis happening. Netosis is abolished. This is how we demonstrated a net formation by X-scan assessment. See here, unstimulated cells, blue color, they are small inside. There is no signal for XCCAT. This is overlay. X-scan assessment. So nucleus is stained. Presence of X-scan assessment. Size is bigger. You can see a lot of signal for XCCAT. This is overlay. This is a positive control PME. Yes. And this is the principal applicable. And this is how PAD4 enzyme presence was monitored. You can see PAD4 unstimulated cell signal is very feeble. PMA, we have a lot of signal with increasing concentration of X-scan assessment, 5 microgram, 25 microgram, 50 microgram. You can see increasing. So this is loading control. So in response to PAD4 activity, we have H3CAT formation. So unstimulated cells, no H3CAT. We have PMA, we have a lot of signal, 5 microgram, 25 microgram, 50 microgram. You see a lot of H3CAT. H3 alone is actually loading control. So these are the corresponding principal time. And don't have to worry about this. See, extensive work by various groups resulted in classification of netosis into two types. This classification is based on the time taken for netosis to happen, and also after netosis whether the cells remain viable or not. Based on these two parameters, there are two types of netosis has been classified. Number one is suicidal netosis, otherwise called delayed netosis. It takes 2 hours to 4 hours. Suicidal netosis or delayed netosis, in this case what happens is after net formation, cells die. That's why it is called suicidal. It takes long time, 2 hours to 4 hours. So PMA 4-bar crystal acetate, cascade bicarbonate, and even high concentration glucose will induce suicidal netosis or delayed netosis. And in contrast, we have another type of netosis. We have vital netosis or early netosis. So this takes 5 minutes to 60 minutes time for netosis to occur. And here, even after nets are released, the neutrophils remain viable. So based on this, we have suicidal and we have vital. So these GMCSF plus C4UA or GMCSF plus LPS, calcium ionophore, cobalamin, and cytokinin saturated from cobalamin are known to induce vital netosis. See, this is what. See, X-scan assessment, see over here, control, 10 minutes, these are almost like 10 minute incubation. They are like control cells from 20 minutes after 30 minutes, no netosis, 40 minutes, no netosis, 50 minutes, 60 minutes, 70 minutes no necrosis. 90 minutes you can see a little release of, let's say, 120 about 2 hours you can see next population. So AT scan would take about 2 hours to maybe it will go on. This is positive. This is co-programmer. Co-programmer, see control here. 10 minutes itself you can see the nuclear state has been altered. 20 minutes they work. Already nets have been ejected. 30 minutes nets have been ejected. 40 minutes nets are almost ejected here. Is it not? So the netosis is completed within 30 minutes itself. And they are viable. I will show you how to viability is. Sir, viability, we have monitored viability through LDH assay. See, 30 minutes netosis has been completed, but no release of LDH. But here, look at this. It takes about 120 minutes to increase netosis to happen, but at 10 minutes, 30 minutes, 60 minutes itself, LDH decay has taken place. So that means, so viable cells generally do not release LDH. Only dead cells will release LDH. So thus, the Naja venom induce, like cobra venom induce vital netosis, so the Leakey Canada venom induce suicidal netosis. So mechanism of netosis is highly complex. You have two types of netosis, suicidal netosis and vital netosis. It takes 2 hours to 4 hours, and here it takes 5 minutes to 60 minutes. It is ROS dependent. All these enzymes play. ROS dependent. So in this case, mitochondrial ROS may play a role. It's NOx, neutrophil elastase, myeloperoxidase, PAT4 not required here. Example, these would induce this. And here, eject nuclear DNA, and in this case, eject mitochondrial DNA as well as nuclear DNA. So, for ejecting mitochondrial DNA, it is ROS dependent. For ejecting nuclear DNA, it is ROS independent, where NOx enzyme, that's PAT4 enzyme play a role. So the same thing I have summarized here. So even lifestyle disorders like diabetes, diabetic gangrene, autoimmune disorders, cardiovascular disorders, neurodegenerative disorders, cancer, in all these cases you can see the role of necrosis. So necrosis is something like double-edged sword. It could be beneficial to clear microbes, or it could be even harmful in these lifestyle disorders. So in summary, necrosis is an ultimate defense offered by neutrophils. Both infectious and lifestyle disorders induce necrosis. Both particulate and soluble factors induce necrosis. The mechanism of necrosis is complex and complicated. Understanding the physiology of necrosis is exciting. The discoverer is yet to get the Nobel Prize. So I think Walker, Wakeman, after sessions yet to get the Nobel Prize, because it's important mechanism, because necrosis, apoptosis, for which Nobel Prize was given, for this again is an important one. Nobel committee has to identify. So this is my group. I got my PhD Professor Veeran Goda. Before that, I trained under Professor Sastri, and he was my senior, Dr. Rambha Sheshwar, senior in IAC, and he was the ex-director, CS chairman, funding. Thank you very much.
Topic is open for discussion. If you have any queries. Sir, one thing. Statins. Statins are used for treating hypertension. That also induces necrosis.
Yeah, next one.
Statins are useful or
I really don't know, it's difficult to comment on that, but that's what it requires more work than
immunity is lost?
it requires more work. But they have demonstrated statins are useful. Even molecular systems also using the process.
Questions, please.
It's open for discussion. You can ask any questions.
ಕ್ವಶನ್ಸ್ ಈಗಷ್ಟೇ ಬಿಗಿನಿಂಗ್ ಅಲ್ಲಿ ಓದ್ ತಗೊಂಡಿದ್ರಿ ಯಾವ್ದು ಕ್ವೈರಿ ಮಾಡ್ದಂಗೆ ಕ್ವಶನ್ ಮಾಡ್ದಂಗೆ ಒಪ್ಪಲ್ಲ ಅಂತ.
Is there any analysis for instead of PM in order to activate the acidic kinase C? Analysis for PM is in order to activate acidic kinase C.
Protein kinase C activity, it is activated by natural activators like glycerol and calcium, only two. That is a result of phospholipase C activity. PHR level. That apart, only PMA is used for phospholipase.
Ranodine.
Okay.
Ranodine.
Ranodine, yeah, it's a calcium and channel rather. Ranodine. Probably that's not a very well known or explored activator application. Ranodine, yes, we have calcium activated calcium release. That mechanism, ranodine play important role. So it blocks the calcium release. It inhibits calcium release. So again, ranodine is, I think it's an alkaloid isolated from ranodia, some species. I think I forgot it. We have calcium release. We have IP3 receptor is there. When IP3 comes and binds, IP3 receptor open take place, calcium exflux take place, and that is followed by ranodine receptor. Ranodine receptor opening take place and calcium release take place. That can be inhibited by ranodine. Ranodine will inhibit. But as such, I don't think it activates it. What made you ask this question? Ranodine, does ranodine is known for activating efficacy?
Yes.
Is it activating? People are using that.
Yes, sir. It serves as a ITP signal.
So that's what ranodine blocks calcium release. Blocks calcium release or what? Ranodine. It binds to ranodine receptor. Ranodine binds to ranodine receptor and it inhibits calcium release. It inhibits calcium. To the best of my knowledge, it inhibits calcium. Hello, sir. Ranodine is inhibitor of calcium release, right? We have ranodine receptor and the IP3 receptor. Both are there in endoplasmic reticulum. IP3 will cause the release of calcium, while ranodine will inhibit calcium binding to ranodine. That's what.
Thank you, sir.
Any more?
Thank you. Thank you.
Thank you.
Token of economic registration for sir. It's my pleasure to present vote of thanks this occasion. As you all know, it is a special day for us, especially people who are working in science. So, sir already explained the importance of Science Day, why we are celebrating Science Day every year. So, this Science Day, again, important reason for celebration is to create awareness and recognize the contributors and recognition of the real contributors of science and conducting the important events like workshops, seminars, special lecture programs to inspire the young scientists to lead research and involvement in the research. So really, Professor Kemparaju inspired us by explaining or by presenting science behind neutrophils. So really we inspired this your contribution and your dedication to science and the work with the nature of the neutrophils and the blood related biochemical research. And thank you, sir. Thank you for accepting our invitation and inspiring us, our students. Special guest. Thank you. And also, I extend my sincere thanks to our department chairman and senior professor, Professor Kumar sir, for their support and encouragement for arranging this type of function in our department. Also, thanks Professor Dr. Purnima madam, and also I extend my thanks to Vivek and the Vivek family members, all the research scholars, teaching staff, and non-teaching staff, and my dear students. Thank you. Thank you.